Hello everyone. Today in this video I am going to tell you about phagocytosis. Phagocytosis is an immune mechanism performed by the special type of cells. They are macrophages, neutrophils and monocytes. Before entering into the topic, first know meaning of the word phagocytosis. Phago means eating, cyto means cell, osis means process. Total meaning of this word is cell eating process. Whenever a microorganism enters into the body, these phagocytes will attack that microorganism and they will eat that microorganism. In the sense, they will kill that microorganism. So this is the meaning of this word. So let's see how this phagocytosis will take place inside our body. Phagocytosis is initiated by the chemotaxis process. Before this chemotaxis, let's see what is happening inside our body. Whenever there is a damage to the tissue, whenever any damage happens to our body tissue, the damaged cells will release some special type of chemicals like this. These chemicals are complement proteins or interleukins or prostaglandins or leukotrienes, etc. So whenever there is a damage to the tissues or any cells, the damaged cells will release this type of chemicals. These chemicals will result in the attraction of neutrophils or monocytes or macrophages, which will perform the phagocytosis. So when these chemicals are released, our phagocytic cells will move towards the damaged site. So in the same site, we can also find microorganisms. At the same time, whenever there is a damage, the microorganisms will also get a chance to enter into the human body. So here you can see it is labeled as pathogen. At the same time, when injury has happened, the damaged cell will release the chemicals, and at the same time the microorganisms also will enter into our body. What they will do? Once the microorganism has entered into our body, the damaged cells will release the interleukins or complement proteins. That complement proteins will attach to this microorganism. Once that microorganism is coated with the specific antibodies or complement proteins, that is recognized by the phagocytic cells. You can see in this picture, these phagocytic cells are having a specialized enlarged structures which are called as pseudopodia. I will show you. Here you can see these structures. These finger-like projections are called as pseudopodia. What this pseudopodia will do is, this pseudopodia will encircle the microbe and they will internalize that microbe and results in the formation of phagosome. This particular part is called as phagosome. Once phagosome is formed, this phagosome will combines with the lysosome. Here this phagosome is combining with the lysosome and it is forming the phagolysosome. Phagolysosome, like this step. Once phagolysosome is formed, inside the phagolysosome, the microorganism is killed with the help of two mechanisms. One is oxygen dependent mechanism and another one is the oxygen independent mechanism. That means microorganism inside this phagolysosome is killed with the help of oxygen and without help of oxygen. So let's first see how oxygen dependent mechanism will kill the microorganism. Inside this phagolysosome, once the microorganism is internalized, and the internalized phagosome when combines with the lysosome, it is called as phagolysosome, and that phagolysosome will increase the production of oxygen free radical species. Will increase the production of oxygen free radical species, and they are like hydrogen peroxide and hydroxyl radicals and superoxide ions. Like this myeloperoxidase enzyme and chlorine ions which are present inside this phagolysosome will produce this oxygen free radical species, and this will result in the death of this microorganism. And when it comes to the oxygen independent killing, so enzymes like lysozymes and hydrolytic enzymes and interleukins and TNF, tumor necrosis factors, they will kill the microorganism in the absence of oxygen. So in the presence of oxygen and in the absence of oxygen, the microorganism is killed inside the phagolysosome. In addition to this, phagocytes will produce the cationic peptides which are called as defenses, and these are rich in the cysteine. And what they will do is they will form an ion permeable channels to the bacterial cell wall. As a result, bacterial cell wall will get damaged and the cell components will be released out, and it results in the death of this microorganism. And once the microorganism is dead, at that degraded particles will be removed from the cell, and that particular process is called as exocytosis. See here, you can see the microorganism is made into pieces and that pieces are removed from the cell, and this process is called as exocytosis. So this is about phagocytosis. Hello everyone, if you have not subscribed to our YouTube channel Medicology, please do subscribe and click the bell icon so that you will get the notifications of our videos and you can watch our videos first. If you have any doubts regarding the medical topics, please do comment and I will make the video as soon as possible. And please do like the videos of our YouTube channel and share the videos and keep supporting. Thank you guys.